Right.
So our main objective for this project was to find out the caloric content of our assigned baked good. So to do that, we had to determine first what cyclohexane aqueous solution was most successful to separating the fats from the rest of the thing. So for that, we used two solvent tests, and then we used the biuret test to see if the fat samples that we had left over still had starches or proteins in them, which were like the other two main parts of the baked good. And then we had to calculate the caloric content, of course, with the information that we were provided about the calories in proteins, starches, and fats. And then we had to calculate the extraction factor to make sure that our results were accurate. And then for what we said the sample was, we said it just looked like a baked good. We couldn't really come to a conclusion just from the experiments, but it had chocolate chips in it visibly, and so we said it could be kind of like a muffin or something like that.
So our raw data, we were about 0.75 grams, a little off, but close. For our three tests, we decided to do 2.5 milliliters, 3 milliliters, and 2 milliliters. And all of our data was a little not quite as close when we got to our bone mass and isolate. The second and third attempt were pretty close, coming around 0.82 and 85 grams, where our first one was a lot more at 1 gram. And the vegetable oil was also about the same for everything, except for the control isolate again, the first attempt was a lot higher than our second and third attempt.
So as previously stated, the first attempt was a lot higher than the other two attempts. We think maybe we did something wrong in the experiment or in the calculations. And so all of the results were similar for the second and third attempt, with the unknown isolate being less than the mass of the control isolate, except for the first one, of course. The unknown isolate was larger than the control isolate.
So for our dehydrated, it was pretty similar as well for everything else again. We decided to do 3 milliliters and 2 milliliters because as stated previously, we thought the 2.5 might have been a little messed up, so we didn't want to have to compare that data. Everything was pretty similar in weights as well. We had our first attempt was for our dry. Our vegetable oil was up at around 1 gram. So it was pretty similar actually, as well as everything was close to 1 gram. So it's pretty similar actually to our 2.5.
And then here were our extraction correction factors that we got from the control with the vegetable oil, and then there were our calculations to figure out what the percent fat by weight is. It again, obviously something we had messed up in the first sample because 73 is like a really big percentage. But then the other two were also kind of far off from each other, more far off than we expected. So for the second attempt we got almost 15%, and then for the third attempt it was almost 25%. So almost a full 10% difference, which we thought was interesting.
Here for our dehydrated, they're also pretty far off from each other. Almost 10% difference, not quite. We have calculations listed below as well. Same as we did for the first one, we just replaced instead of the moist unk, it was the dehydrated unk.
And then here are our caloric analysis. We went off the fact that the fats had 9 kcals per gram, and that the carb and protein both had 4 kcals per gram. So we used that information to calculate the total calories in the serving, which we said was 28 grams based off the information provided in the thing. And yeah, these are just pretty much all of our calculations for that with the more sampling hydrated.
So here are protein and starch test results. We found them all to be the same no matter what. So all for the positive, the DO water layer is slightly tinted purple, whereas the top cyclohexane layer is a slightly pure yellow. It indicates the presence of protein. As well as the negative control, the layer was mostly white, a little bit of a blue tint. The top cyclohexane layer was a was slightly through yellow. The results were kind of questionable, but it still seemed to indicate the lack of protein.
We basically just got negative results for all of our tests, which showed that our tests were successful because there were no starches or proteins left in the fat samples. And then basically the same thing we got for the starch tests. Like I said, we got them all negative, and we also kept the positive and negative controls the same because they exhibited the same results. When the sample was positive, it had a black layer at the bottom, which indicated that there was starch in it, and none of our samples ended up having that black layer at the very bottom. So we figured there was no starch left in the fat samples.
Okay, for our raw data results, around the mass was the same, the 0.75 grams, and then we used the volumes 2.5 milliliters, 2.0 milliliters, and 3.5 milliliters. And then our mass or unknown was pretty similar to the boat mass. And then the same thing for vegetable oil, it was pretty similar, but more higher than the first. And then, so we found that they were pretty low, 0.58 grams for the first one, and then 0.021 grams for the second one, and then 0.097 grams for the third one. And we noticed that multiple times there have been like remains we were supposed to be evaporating the cyclohexane, so that's why it kept getting more towards the last one.
This is for our dehydrated data results. We found that first when we had weighed our sample, we got to 0.752 and 0.748, so that was as close as we could get to 0.75, and same thing for the control to get to 0.25. And then for our unknown isolate and our control isolate, we saw that they were also pretty low. The control isolate was a lot higher than that. These are our extraction correction factors and percent by weight calculations. So how we did it to get the extraction correction factor, we took the original mass of our control, which was the oil, and then we divided that by our control isolate, and then we took that value and multiplied it by our unknown isolate mass, and then we took that number and we divided it by the original mass of our unknown, and we multiplied that by 100 and took this percent that by weight. We went through the same process to get the extraction correction factor and percent by weight for dehydrated honey, changing the calculations. And in order to calculate our kilocalories, to get our carbs plus protein mass, we took the original mass of the sample and then we subtracted that by the N fat, and then we took that number to convert it into kilocalories. So we took the N fat and we multiplied that by 9, and then we took the carbs plus protein mass, the original value we got, and we multiplied that by 4, and then in order to get our calories per serving, we divided 28. Or I'm sorry, for the kilocalories, we took those two values and we added them up together, and that's how we got our kilocalorie sample. And then we took 28 grams and divided that by that number in order to get the calories per serving. And we basically followed that same step for our dehydrated sample, but we did a moisture correction factor. So we took the mass that we weighed in the watch glass mass after it was dehydrated. And the same steps we took, except at the end to get the calories per serving, we multiplied 28 by that number of the moisture correction factor, and then we divided that by the mass of the sample and multiplied it by the kilocalories.
And for our protein results, for our positive control and negative control, they're the same. Mostly we've got purple hue on the DI water layer, but the last one we did get all white hue instead. For our negative, we got white hues mostly. One time we got bluish gray hue. And then for the results of the scientific dessert, we found that most of them was clear or white hue at the bottom. And there's multiple times where we had both layers clear. So meaning there's no protein. And then for our starch results, we also found the same for the positive and negative each time, and we found that it was dark red at the top, and then black hue for the positive, and then the negative was orange tint. Throughout the whole results for the assignment dessert, it was also a red hue and orange tint for the bottom layer. But we did have one time when both of the layers were red, but there was no black, so we indicated that there was no starch.
And then this was our just comparison of results between the two groups. So we did get some similar percentages, obviously with our first percentage being really high like we mentioned earlier. And yeah, we got kind of similar results, but some of them were a little different.
In the analysis of the results, we found that the best combination solvent was the cyclohexane and DI water, just because they had more positive results, negative results for both of the tests, meaning there was more clear separation. And then the best solvent volume was 3 milliliters because it had more accurate results. Because for ours was like 2.5 was more accurate, and then for theirs was 3 milliliters. So we just did 2.5. For the moist and dehydration, they both showed similar results for both of us, so we had no correlation between the two. And then for the similarities and differences, we saw that we both changed the solvent to 2 milliliters. And then group 1 believed there was an error made in the first dessert attempt. And then for group 2, the calorie servings were slightly more lower than their group 1. And then we thought that group 1 was more successful because they